It's a pleasure to have the opportunity to talk here. I should start out saying that I'm really not a health researcher. I really know nothing. Just this past year, I attended my first public health class where I actually gave a guest lecture. So I don't know. I really don't do exposure assessment. I say that really I'm an engineer, at looking at things like things that make pollution. And if anything, my second thing is I'm really a chemist. But I would say that a lot of what I do currently and historically have done is looking at how we can use our understanding of engineering, of sources, as well as chemistry to help inform or help bridge the gap in things that we do with health studies. And so really what I'm going to try to talk about today is sort of the kind of tools that I work with and really give some examples of how those tools work, and then with the idea that ultimately we want to integrate those type of tools into things that Mike was talking about and Petrus was talking about before, and these tools are really looking at sort of analytical tools and methods. And so I guess that's kind of where I start from, and I'm not really going to talk about details and methods because most people don't want to hear about too much chemistry or biology, but really just talk about the strategies that we can use about looking at detailed measurements in the atmosphere. And I guess, I guess I have to use this to advance it. So I guess what I'd like to do is just spend a little bit of time talking about atmospheric particulate matter, because for me, a lot of times we think about atmospheric particulate, the mass concentrations that we can look at, but I actually am very interested in the very detailed chemistry of the particles in the atmosphere, and talk a little bit of summarizing my perspective about health effects, and what I'm going to talk about is sort of things that sort of motivate some of the work I'm interested in now of reactive oxygen species, which was mentioned this morning, but this idea that this is one of the pathways related to health, and the idea that what we can do in terms of chemistry and analytical tools to help understand more about sources and chemistry related to that, and then talk about this idea of how we can maybe begin to integrate some of these advanced methods or just chemistry into these kind of studies. And I'm going to give 3 very quick examples of just things that I've done which sort of bring the interface between source apportionment, chemistry, and kind of trying to connect that to health. And that's sort of where I'm going to head. So real quick, I guess I just want to say, and maybe this is obvious, but I think it's important to state that when we talk about atmospheric particulate matter, we obviously are concerned about health issues, but it's these same particles that are really important for things like human welfare, visibility, the damage of materials like crops, and also materials that we engineer. There's obviously major issues about ecosystem impacts. We talk about things like nutrients and toxics, and also these are things that are also very important for climate forcing. So it's the same pollutants that there's a big co-benefits of reducing these pollutants. I just have these 2 pictures here. This is a picture I took. That's the bird's nest in Beijing and the Olympic torch, and this is the Kathmandu Valley. And it's not just a question of, in these cases, of human health, human welfare. These are also huge economic issues. When I was involved in planning for the Olympics, and this is really significant to economic development, because people come here to see the beautiful mountains that you can't see. People don't want to do business in an environment like that. So there's many issues that motivate these, although what I'm really going to focus on and what we're focusing on health, but we shouldn't forget these really important co-benefits that exist from controlling particulate matter in the atmosphere. One of the things that I think is really important to emphasize for people who are not real familiar with particulate matter in the atmosphere, when we talk about particulate matter, in my view, my particulate matter perspective is sort of, it builds upon with the talks that we've heard earlier today, is that there are very big gradients, not only in concentration, but there's major gradients in composition and sources. And so if we look at the chemistry of them, these gradients spatially and temporally are enormous variations. An example here is this is actually a daily measurement of leachable iron in the city of Denver that we measured for a year, and I'll use this data later. But you can see that, if I go out and make a measurement on this day and measure it here, these are drastically different. And we should be aware that things are changing. Here's an example of actually a vehicle in Lahore, Pakistan, and you can see the spatial gradients. And another example here is actually, this is a study in Beijing where you can look at, these 2 samples are a week apart, and the gray here is dust, and these are other secondary pollutants. So within a week, we can have the same concentration, but a completely different composition. And obviously that's important when we think about health. We can't just think about PM or talk about the annual average. These gradients are very important for exposure and also very important for health effects, and I'm interested in understanding about how that chemistry changes. I've done a lot, which I won't talk about today, about how sources change that drive that, but also what does this mean for health? This is just an example. I just had, as a domain, a project that I was fortunate to be involved here. It was funded by Merck, which actually looked at 11 cities in this region and looked at sort of the annual average composition. And one of the things that's interesting is that this is an annual average, so it really buffers the differences. But there's a lot of differences. There's a lot of similarities, but there's also a lot of differences in the bulk composition. And bulk composition are things that we normally monitor. But if we start looking at some very detailed things, for example, here shows some example of actually one of our sites in Zarqa in Jordan. You know, you start talking about these very trace amount of metals that could be 10 or 100 times higher than other places. Likewise, the sources of this yellow, which is organic material, could be drastically different. And one of the studies, the papers that were published, shows that things can be very different. And so again, we want to be aware not just about concentration, not just about bulk composition, but we really want to get even deeper than that into what these materials are. So from my point of view, as someone who doesn't do health studies, I'm aware of the epidemiological data, and sort of the angle that I approach things from is I think it's very clear from things we've heard of before and things that we can see in the literature that, you know, there clearly are important relationships between fine particle concentrations in the atmosphere and health. I think even some of these health studies are beginning to say that we can understand a little bit about the sources and also about the chemical composition, how that relates to health. But for me, I think as somebody who, again, is an engineer who understands about technology for controlling sources and strategies for thinking about fuel supplies and sources, I really want to know more about individual sources. And also as we think to the future of designing control strategies, how we make changes in things like roadways or fuels, and what does that mean for where these particles are coming from and what they're comprised of. And so one of the things that I'm really interested in is this idea that we can begin to look at this detailed chemistry and connect that with biological systems, either through in vitro or human studies. And the ideas that I really want to do is talk about sort of a framework that we've been working on where we can help kind of bridge these gaps. And again, I don't mean to imply that we're going to do an in vitro study and stop doing epidemiological studies and stop doing things, but I think that we can learn a lot about the detailed chemistry and about the sources and what that means, integrating all these kind of tools together. So just as a quick example, I want to, as a study that I was involved with in rural China, which was actually what the answer might be before about solid fuel burning, whereas a student that I helped do some measurements with where we're basically looking at blood pressure and particulate matter exposure from indoor cooking. So this is a single source, but it's in some ways a very well-characterized source. And I'm not going to go through the details of the study, but the bottom line of the study is that we could see blood pressure effects from this exposure. So the question that motivates me is not only is it just the PM, but what about this PM? It turns out this is an interesting study where this was 8 different villages, and it turns out there's a major roadway that goes through these villages, and the risk factor is actually different depending on the distance to the roadway. So even though this is dominated by an exposure of biomass burning, it certainly suggests that we need to know something about other sources. So with that said, it's just an example of sort of my sort of paradigm or picture that I think about is that, you know, people that I work with very often in the atmospheric chemistry, we can think about all sorts of complex chemistry that happens in the atmosphere, the sources mixing, chemical transformations, and likewise there's a lot of very complicated biology. And my experience has been that people who study sort of the toxicology or biology take one sample from the atmosphere and study that for a while, and likewise people here would say, tell me what chemical is important and I'm going to worry about this. And so what our hope kind of is, or the strategies we want to use, is to try to mix these together a little bit more so we can really think about not just the mechanisms here, but how the changes in the atmosphere affect what happens on from a biological point of view. And I want to give some examples of sort of how we would do that. The example that I've largely been working on, and I don't mean to imply that oxidative stress is everything, but it just happens to be something that I've been working on, and I think everybody here is aware of the issues of oxidative stress and how that's related to many of the diseases we're concerned with. And so for us, what we're interested in is actually looking at how the redox activity, or we often call reactive oxygen species of particulate matter, what in the particulate matter controls that, and then ultimately how we can relate that to human health issues. And so we, in our laboratory, have developed, I'm not going to go through a lot of details, but a macrophage assay where we basically can collect particles in the atmosphere. We can incubate them with macrophages it's a rat alveolar macrophage, and we can use a chemical probe to say how much RS is generated. And so what I'm going to explain a little bit is that although people have done this before, it's not something tremendously novel. What we're interested in is actually looking how the particles in the atmosphere change. If you think about that study in Denver about the iron going up and down, but also we can in the laboratory fractionate these samples and we can selectively remove components and then say how does that component affect this biological activity. And so again, I don't mean to suggest this will replace health studies, but I think this could really be a great opportunity to integrate them so we can understand, make connections more to sources, chemistry, and what the health effects would be. I'm not going to go through much of the details of this. I'm sure many of you know more about biology than I do. But one of the things I want to mention is that this assay that I just mentioned were actually used in human health studies. And one of the things that's exciting about it is that when we look at, in this specific study or a few other studies, if we look at things with patients with COPD, it turns out that our ROS assay is a good predictor of circulating biomarkers or exhaled NO. So both for lung inflammation and systemic inflammation. And so for us, it feels like at least there's a connection we can make to human health. So we've done a lot with this cellular assay, and the idea is that hopefully we can rate sources, chemistry to the assay, and then ultimately the assay to humans. So I want to go through 3 quick studies, as I mentioned before, and just sort of talk about sort of this idea of how we can connect sources to chemistry to this biological activity. And so this study in Denver, which I showed the iron before, we basically collected samples every day in Denver for a year. We measured bulk chemistry, like I showed in those graphs before, but we also measured water-soluble elements. We measured 52 elements by ICP-MS, and we took 50 randomly selected samples, and we did this ROS assay, where we looked at the biological reactive oxygen species for a macrophage assay. And so I just want to show some examples. This is an example of a sulfur, and again, the point I'm making is if we took one sample instead of here or one sample here, we'd have a very different answer. And the idea is that we want to understand how the sources vary and what's important. So sulfur is here as both is also sulfate as a secondary pollutant, but it's also related to primary. Some of these peaks are due to plumes that come into the sampling site. This is leachable potassium as an example. If you can tell, this is July 4th. I think everybody knows that's from fireworks. And so, example, I don't want to conclude that fireworks are what are killing everybody, but the point is things vary a lot. There's a lot of complexity in the atmosphere. Here's an example of the iron. And iron is important to us because it's very redox active, and it turns out something very important in this study. And I could go through many, many graphs, but I'm not going to. What we did is we used some statistical methods to understand what factors drive these concentrations, or so often we call this source apportionment. We use something called a positive matrix factorization model. We're saying these are factors. Factors are driven by atmospheric processes as well as sources. And then we did a multivariate regression to look at this ROS, how we could explain what that activity is. And just real quick, this is a map of the different sources we could identify. And again, I really call them factors. You know, there's a lead point source, there's iron sources, dust, and different things we could look at. But what's important is that we then wanted at the end to sort of say what is driving this biological activity, and we could look at that. And it turns out 3 of the sources, this water-soluble carbon factor, which is with sources we understand in the atmosphere, resuspended dust, and also this iron source are what drives this biological activity. So again, it helps us direct where we want to be looking for what these sources are, and as we can inform health studies that these are things we probably might want to be looking at. The next study is actually a crazy part of the world that we've done studies in. Is the picture I showed before in Lahore, Pakistan. Fortunately, we're not doing any more work in Pakistan. It's not a great place to be. I never went there for the study, but we are still doing stuff in Delhi, which is in Agra, which is very close by. Same kind of issues. These are we ran a similar kind of study where we actually looked at the same kind of chemistry we do every 6th day in this case, and we looked at our cellular activity, and we also looked at different types of metals. And in this sample, what I'll say briefly is we actually did the fractionation, where we selectively removed metals to see what that meant for the biological activity. This is a part of the world where I do a lot of work in China, and I tell people in China, I like to present this data in China because it makes people feel good about their air pollution. The PM10, PM2.5 levels are incredibly high, but what's even more staggering is not that the levels are that high, but the composition of the PM is sort of mind-boggling. Here's a snapshot of this is the PM10 concentration, and the units are right, micrograms per cubic meter of lead and zinc and other metals. I mean, we're talking, where I live in Madison, Wisconsin, our PM10 levels are not even as high as this. I mean, they're incredibly high metal concentrations, so this PM is clearly different than PM other places. And I won't show the data, but this is true for PAHs, it's true for many, many, many different components that we should be concerned about from a health point of view. And so again, we can look at things like the seasonal trends of what this ROS activity is. We can look at the difference between PM2.5 and PM and the coarse PM, which is PM10 minus PM2.5. We can look at the trends, and we can relate that to what the chemistry is. What I want to just emphasize here is that if we look at this is actually the biological activity relative to mass of PM, so this is much higher concentration, much higher activity or activity than we would see in a lot of the sites we've looked at in the US, and the concentration is much higher. So given that, we are really interested in understanding what is driving that. We can do we did a similar thing like we did in Denver, where we could do statistical methods, but for us, as many people know, association isn't really shows the causality. So in this study, we actually did some work where we took the samples and we chelated the metals out. So we could selectively remove different metals by chemistry and then do the assay again. And so in this case, this is a siderophore that's actually selectively for iron. And what we can do is we basically looked at the biological activity. And what's interesting here is that this is the original activity in dark for different times of the year. And we can look at what, if we only remove iron, what that affects the biological activity, and if we remove all the metals. And I'm not going to go through all the statistical results here, but the message here is that iron seems to be important from a mechanistic point of view, but the activity we see is not proportional to iron, it's proportional to other metals. So iron, the absence of iron is important to the process. If we take iron out, not that we use iron as a control strategy, but the idea is that it gives us an understanding of what sources and what components are important to what's going on. Another example of that, the final example, is actually from a study we did in Long Beach, and Long Beach, California, the LA area, because this actually, what's interesting there is that we see that this ROS activity is highly related to nickel and vanadium coming from the port. And so what's interesting is that if we move farther into the LA area, we see other sources dominating this biological activity. And there's been a lot of studies recently showing about how the dispersion of the ship emissions are and how they affect things. And so what we did in this study, though, was actually, we're sort of advancing, and this is sort of where I'm kind of lead towards the end of my study, is we actually decided to expand and not just measure this ROS activity, but we're actually doing a gene expression assay where we basically look at oxidative stress indicators and markers, very similar to the ones we looked at humans. And the idea would be is we can look and see how these particles affect, when they're with the macrophage, this sort of signaling that goes on. And I'm not going to go through the details. We have toxicologists that we work with that sort of work a lot of this out. But the idea is that we can see a couple quick things. I want to show some snapshots of this data. Is that this actually shows, if we took these samples and we took only the water-soluble components versus the insoluble components, and we understand what the sources of those are, we see that the ROS activity is actually not so different. But if we look at the TNF-alpha signaling, which is important for sort of systemic effects in the body, we see that the unfiltered is what drives the activity in almost all cases except one location. So again, it's a way that we can begin to think about what sources are more important. This is another example where we can actually, and I'm not going to go through the details of this, but we have a filtered sample, which is basically after filtration, and we do all these different kinds of chelexing to remove selective materials. The C18 removes organics, these remove different metals, and you can see what this means both to the TNF-alpha, the ROS, as well as this other chemical sensor of these. And so the idea is that we're really trying to understand how we can fractionate this out from a source and chemistry point of view and relate to these biological processes that would be human to health. And so again, we see this sort of a linking between the studies we do with humans and what's happening with sources in the atmosphere. So really, I just want to conclude by saying that in my opinion, there's a great incentive to bring these biological assays, but also linking them with our ability to understand the chemistry and sources into these health studies, and depending on the design of the health study, they can be integrated in different ways. These assays that we do become pretty simple and pretty standardized. They're not that expensive to do once you have them up and running. And I think there's a tremendous opportunity for us to understand about sources and source apportionment again, ultimately, in my opinion as an engineer, if we start talking about developing control strategies, we really are going to control sources. We don't just say we're going to control PM. We have to say, what are we doing for this technology or that? And there's always trade-offs. So for us to be much more tailored is important. And again, I think that there's a lot of opportunities not to just look at statistical associations, but we have there's historical records of things of mutagenicity, which I'm not doing mutagenicity, about manipulating samples, and there's a lot of tools today that we can do that can really help these kind of studies. And so with that, I just acknowledge all the people and the funding that have been involved in these studies, and I'd be happy to try to answer any questions if there are any.